Okay, welcome back. Hope everybody is doing great today. In Chapter 7, we will be focused on viral detection and diagnosis. We'll start by talking about what types of samples might be collected in order to perform some of these different tests. Um, we'll talk about how you would culture a virus in a lab setting, which basically means having to grow cells that would be the host for those viruses, so cell tissue culture. And then we'll go through some of the different types of common tests. And so some of those are going to be tests for viral antigens. Some of those are going to be tests for antiviral antibodies, or at least will utilize antiviral antibodies. Some of them may be testing for the presence of the viral genomes directly. And I will have this divided into two parts, two videos. All right, so it's estimated that at least 60% of infections that doctors see are caused by viruses. So it makes sense, if the majority of infections are virus infections, we would want to be able to identify the viruses. And of course, there's quite a few reasons for needing to have a good ID. In some cases, if it's just common cold, maybe it's not that critical. But it's hard to prescribe the best treatment if you don't know what virus you're dealing with. And this is complicated by the fact that a lot of viruses, especially initially, may present with the same set of symptoms, right? Fever and achiness and sore throat, some of those kinds of things. But if your patient is in a high risk group, it would be potentially life-saving to know exactly what virus you're dealing with. If you're thinking about providing safe organs or blood for transfusion and transplantation, you would want to be able to screen for an assortment of different viruses. So I also just wanted to point out up front that a lot of these diagnostic tests that we're going to talk about in this chapter are also used all the time in research laboratories. So virology research relies very heavily on these tests as well. So let's start by talking about what kind of samples would be utilized for these various tests. And of course, it depends on the virus and what the portals of entry and exit for those viruses are, what tissues in the body they preferentially infect. But you can see from this table, right, very often it may be serum. So serum is the liquid part of blood. Basically, collect a blood sample and spin out the cells. Or very typically, some sort of basically, nasal mucosa or genital mucosa, saliva. Again, it just depends from virus to virus. And we're all familiar with the nasal swab now from COVID-19. But in all these cases, as I mentioned, some of these tests are going to be specifically trying to detect the virus itself, whereas others may be testing for the presence of antiviral antibodies in the host, which would indicate a specific infection as well. And so we really have these three general approaches to diagnosing viral infections. We've got tissue culture, we've got serology, and then we've got assays that are basically molecular, something like PCR, for example. So I'll go through these one at a time, and I want to start here with tissue culture, aka cell culture, which is referring to the ability to grow tissues or cells outside the living body in that laboratory setting. And so, because viruses are intracellular parasites, if you want to propagate viruses in the lab, you have to propagate the cells that they would be capable of infecting. And this will allow us to look for things like cytopathic effects, which are basically visible effects that viruses may have when they infect cells. Also lays the groundwork for cellular assays, and I'll give you a couple of examples of commonly used cellular assays. And this tissue culture approach contrasts with serology, which is tests that would utilize serum. And then I already mentioned molecular biology would include things like PCR testing. All right, so if you're not familiar with the idea of cell culture, you can grow human and mammalian cells in the lab in petri dishes or 96 well plates or tissue culture flasks like these, and you can grow basically monolayers of cells on the surface of that plate that will look something like what you see in these two images, with slight variations depending on the particular cell type. But in these assays that utilize cells, again, we basically have either direct or indirect tests. So in a direct test, you're actually looking for the presence of the virus itself, whereas in an indirect test, you're looking at the effects, maybe for specific effects that a virus might have on those infected cells. And those would be those cytopathic effects that I mentioned previously. So here's some examples of the indirect cytopathic effects. This is human herpes virus 6, will typically cause these ballooning of infected cells. You can see indicated by the arrows here. Right, that's a very unusual phenotype that's pretty specific to this virus. I'll point out this one here in SARS and COVID-19 infected cells. You can get these syncytium, which are these big viral structures that are very obvious via light microscopy. So, not all viruses are going to give you obvious cytopathic effects, but some will. And, a particular cytopathic effect might not necessarily be diagnostic of a specific virus, but it would at least help you narrow things down. And in some cases, it might be more definitive. So we're taking these cells, taking these tissues from the patient in some cases, and we can do things like cytology or histology. You're probably familiar with those terms. The difference between these is cytology is you're actually growing cells from a patient sample in the laboratory setting, whereas in histology, you're taking a biopsy and then you're just looking at that tissue. So it's probably going to be a fixed, maybe stained sample, but you're not actually growing the cells in the lab in histology. And as I just said, oftentimes you're going to have to do some kind of staining or labeling just to enhance contrast and allow you to see specific parts of the cell better. And so before I get into some of these cellular assays, another important thing to mention is, anytime you're working with virus or virally infected cells in a lab, you want to take proper precautions. And so we have these well-established biosafety levels, BSLs, that specify specific precautions depending on the risk level associated with the pathogen in question. So we have biosafety level 1 through 4. And just to give you some feel for what the difference between those 4 levels is, biosafety level 1, you're working in a lab with minimal precautions. So this would be for very well studied viruses that don't cause disease, or definitely don't cause serious disease in healthy adults. There's very minimal risk. Researchers are going to be using gloves, maybe lab coats or safety glasses if necessary, but you don't need a biological safety cabinet. That would be like a hood, unless you're using one just to keep your cell culture sterile, which would be pretty common. But this would be the case for most animal viruses that may not even be capable of infecting humans. So just your standard lab setting is appropriate there. For biosafety level 2, these would be viruses that pose moderate hazard. Um, and in level 2, you have those same precautions as in level 1, but there's going to be some supervision, some additional training, and it's likely that you would probably be using a biological safety cabinet. So this would be if you're working on hepatitis or HIV infected cells, for example. In level 3, now these are going to be potentially lethal viruses that could be transmitted through inhalation. So here you're going to have all the same precautions as level 2, but you're going to have to actually have a laboratory that's been designed for this specific purpose. So there's going to be 2 self-closing doors under negative pressure. It's going to prevent air from escaping from the lab. All the air in the lab is going to be HEPA filtered. There's going to be more rigorous PPEs being used, might be full suits in some cases, masks. Definitely more serious in this case. This would be something like SARS or West Nile. And then the highest level is level 4. So these are going to be, extremely dangerous exotic pathogens, something like Ebola, that are definitely going to be life-threatening. In this case, researchers are going to, be in specialized rooms with filtered air, but they're going to have to completely change out of their personal clothing. So basically, nothing that goes into that research environment is going to leave, right? Researchers are going to be wearing essentially spacesuits. Everything will be completely contained. All right, so we got those 4 biosafety levels. So let's come back to the tissue culture, and I wanted to just show you a couple of examples of what we're going to call cellular assays. And so what we've got here is a list of some very common diagnostic tests for viruses: immunofluorescence assays, immunohistochemistry, enzyme immunoassays or ELISA, Western blots, called agglutination tests, lateral flow immunoassays. And really the first 2 would be considered cellular assays because they are using whole cells rather than serum. And I would add a third to that, which is what's called the plaque assay. So I'm going to show you what these 3 look like: immunofluorescence, immunohistochemistry, and the plaque assay. And some of these are going to take advantage of engineered antibodies that have been specifically designed and are really being commercially produced to identify specific virus. So that these antibodies are being used as a tool that is part of these specific tests. And I'm saying they're engineered because a lot of times they're what are called conjugated to a dye or a fluorescent molecule or an enzyme, something that's basically going to allow you to see where that enzyme is or where the antibody is located. And both of these tests are going to utilize antibodies, so you'll see what that looks like here in a second, these first 2. So let's take a look at these, and we'll start with the immunofluorescence assay. And so again, we're looking at cells, right? So these could be cells that are being grown in culture, or these could be cells from a biopsy. Just depends on the particular situation. But the way this would work is, so we've got an uninfected cell here on the left, infected cell on the right. The infected cell has some viral antigen, right, present on the surface of the cell. And we have these engineered antibodies that would specifically bind to that viral antigen. And as I was just talking about, these have been engineered to, they've been conjugated to a fluorophore. So this is a fluorescent molecule. When you shine the correct wavelength of light on it, will literally glow in the dark. So if you have an uninfected cell, these antibodies won't bind and they just get washed off during the assay. Whereas if you have an infected cell, the antibodies stick, and then when you look at them under a fluorescence microscope, again with the correct wavelength of light, you will get something that looks like this, where the infected cells, you're seeing that fluorophore glowing very brightly. And so this, let me just go back, this works if the antigen is on the cell surface, but this approach can also be used in cases where maybe you have a virus that isn't producing an antigen that's being expressed at the surface of the cell. Everything's internal. There are some tricks where you can permeabilize the membrane and let the antibodies actually get inside the cell to bind to the antigen. All right, so that's the basis of immunofluorescence assays, right? The immuno is because we're using that antibody, the fluorescence, obviously, what we're using to visualize. And I wanted to point out this distinction as well, because this will come up in some other tests. You have what's called direct immunofluorescence or indirect. And we just talked about direct, where the antibody that binds to the antigen has that visible fluorophore directly attached to it. In some cases, it might not be possible to design antibodies that bind to the viral antigen that also contain that fluorescent marker. So in that case, they'll use this little trick where the primary antibody doesn't have any label on it, so even though it goes in and binds to the infected cell, you can't see it, but you can actually generate antibodies to other antibodies. So you put in what's called a secondary antibody that now has that fluorophore on it. So now you can see where the primary antibody is located because the secondary with that tag is sticking to it. So it's a little bit of splitting hairs. It's just an extra step in there, but the end result is the same, right? The infected cell will glow. You just need 2 antibodies in this case instead of the 1 here. Okay, so what about immunohistochemistry? That was the second type of test. It's essentially the same idea, but the difference is the antibodies, instead of being conjugated to a fluorophore, are conjugated to an enzyme that, in the presence of the correct substrate, will produce some kind of colored precipitate usually. So the antibody binds. You would then wash away any antibody that does not bind, add this particular substrate, and you'll get this color change that is going to let you see where the antibody is located. And so, again, it's basically the same idea, but you don't need a fluorescence microscope in this case because it's an enzyme-based test. So you could just use a standard light microscope to actually be able to see where your antibody is, where those infected cells are. So here we're getting this red precipitate everywhere that there's antibody bound to viral antigens. And then the third one I wanted to mention was the plaque assay, and this is very commonly used. So a plaque is basically these little clear circles that you would get on a monolayer of growing cells that shows, it's basically a circle of dead cells, shows where a viral infection occurred. So imagine here the red is your cells being grown on these plates, and everywhere a virus infected a single cell, right, it spreads to the neighboring cells, and you get this little circle of dead cells. So you can see the clear circles here on this red plate. So each one of those circles would be a plaque. And depending on the number of virions that were present when you infected that plate, you'll get more or less plaques, right? So here we've got basically 3 different dilutions where on the left you've got just tons of plaques, right? There's barely any red left, any cells left. So this, you can do calculations to come up with what are called the number of plaque-forming units in the inoculum, right, in your sample, and that's basically an indication of how much virus is present in that sample. So this is very common. You'll see like plaque-forming units, PFUs per milliliter, and it's just a measure of viral concentration, essentially. Okay, so those were some of our cellular assays because we were looking for virus in cells. Most of the others on this list are serum-based assays, so that's going to transition us into the second half, and I'll pick up and talk about some of those assays in the second video.